Hello. We are going to be continuing our discussion about making a protein, and we are going to be starting with the mechanics of transcription. So remember, we left off the other day, we left off with this question: how do our cells know how to build this amino acid strand versus this amino acid strand? And those would be our different proteins. So we've got hemoglobin proteins that code, that are for storing oxygen. We have proteins for transport. We've learned about proteins that are enzymes. And these proteins are made of amino acids. So, how do our cells know how to build these? And so that's what we ended with on the other day. We understood that the DNA codes for amino acids, and the codons would be three base pairs. And so you learned how to use the codon chart: methionine, proline, proline. These are all different amino acids. And how do we find them? We use our first letter, second letter, and third letter. So just as a quick reminder, that's what we learned the other day. So it's the sequence of mRNA that then dictates what our amino acids are. So if we change our order of our nucleotides, instead of AUG, if we had AG, then we're going to change what our amino acid is. And that becomes very, very important. Now, the process of transcription is actually a pretty basic one at the level that we're going to be looking at. Basically, we want to open up our DNA, and we're going to use one strand as a template strand to make the copy. Now, this template strand could be either strand of DNA, and in fact, for some genes, it's the one on top, and on some genes, it's one on the bottom. And so we open up the DNA to get access to our gene. And then we have an enzyme called RNA polymerase. That enzyme, RNA polymerase, is the one that actually makes the RNA strand, connects them together. Now, don't overthink this word. RNA polymerase makes RNA. DNA polymerase makes DNA. They've made it that simple. So RNA polymerase is the enzyme you can see down here in this picture. It's the enzyme that stitches together our codons. And then we're going to get our start codon, AUG. Now, this picture doesn't have an AUG, but a lot of our genes start with that start codon. So here it is, just in another diagram. Once we have it started and initialized, then we just elongate. We're going to make our mRNA strand from our template DNA. Now, different diagrams show different ones being used. This one's showing the bottom one. Sometimes we see the top one being used, but it depends upon what side is the template. But the end result is we're going to have a long strand of messenger RNA. Now, we can make other types of RNA in addition to messenger RNA. But the messenger RNA is what contains the code to make that amino acid sequence. We can make other types of RNA, and transcription will make them. One of the first ones we'll look at is ribosomal RNA. Now, ribosomal RNA really does not look like much. In fact, it looks right now like a big tangled mass of yarn. Imagine, if you will, a long piece of RNA that doubles back on itself and makes these connections, like a big ball of sticky tape that folds back, and then you make this mess of sticky tape. Well, that's kind of what ribosomal RNA does. Now, the only thing I really want you to know is ribosomal RNA makes up a ribosome. That is its goal, that's its job, is it kind of makes the structure of a ribosome. But how do we get it? Well, we actually transcribe genes to make that ribosomal RNA. Now, the other RNA that we're going to look at, and we're going to unpack this a little bit, is transfer RNA. Now, transfer RNA, again, is a long strand of RNA, and it folds back to form this kind of goofy looking clover leaf that kind of bends. And so you can see different books have different diagrams that kind of show it. But here we have our mRNA, and the job of tRNA, transfer RNA, is, as the name implies, we want to transfer an amino acid. And so you can see this kind of goofy clover leaf shape, but in reality, it kind of bends to the side. And so here we have a long strand of RNA that kind of coils up on itself. But here's the most important thing. One end of the tRNA contains 3 bases, 3 nucleotides, and what they're going to do is they're going to connect to a codon. And so we call the one on tRNA an anticodon, and it's going to connect to an mRNA codon. And notice they complement each other. So right here where my pointer is, the CUC, that's the anticodon, and then the GAG, that is the codon. So the mRNA contains the codon, the tRNA contains the anticodon. So one end of the tRNA contains an anticodon. The other end of the tRNA contains an amino acid. And here is the big takeaway. The anticodon will specifically dictate what amino acid that tRNA carries. So the tRNA, its job is to act like a translator. It speaks both languages. It has the codon language, it matches up with the codon, and then it carries the right amino acid with it. And so in that way, you're going to get a specific tRNA for each amino acid. So notice down at the bottom, we've got a tRNA that has a anticodon UAC, and that anticodon UAC will fit the codon AUG. And it's going to carry the amino acid methionine. And then the tRNA UUA, that anticodon will match the codon AAU and carry the amino acid asparagine. And then the tRNA that carries the anticodon GUG will match the codon. Well, let's see if you've got the idea now. We've got the anticodon GUG. What would base pair? What would be the complementary base pairs for GUG? Hit pause and see if you know. All right, hopefully as a class you decided that if the anticodon is GUG, then the codon should be CAC. Yay, good job. So that is our tRNA. Now the last thing I want to discuss with you would be our the starting point of our translation. Now, there's a lot on this page to unpack, but let's start with the idea of what do we need to translate? Well, we need the actual message, right? We need the code. That would be the mRNA strand. And then with the code, we need a code breaker. And that would be the tRNA. The tRNA is what speaks both languages. We're going to bring the right amino acid to the mRNA. And then we need a place for all of this to happen. So in this case, it's going to be the ribosome. Now, 2 months ago, when we learned ribosome, all we needed to know is that a ribosome makes proteins. Well, now we can actually unpack these, and we know that a ribosome is going to connect amino acids together. And it doesn't look very exciting. It's this big brown lump right here. That's the ribosome. It's got a large subunit and a small subunit, and then it's got different areas on it, and it's got docking areas 4 different places. So let's take a look at these. What we end up having is a space where the mRNA is going to line up, and that's going to be our code. Remember, the mRNA is our code. And so, along with our mRNA, we've got to have spaces where we can bring the right tRNA. And so we're going to have the right tRNA come up and bind to the right codon on the mRNA. So let's take a look at this. In our A site, we've got a new tRNA with the appropriate amino acid that comes into that spot. In the P site, we've got a tRNA with an amino acid sits in the P site. These amino acids have already been joined to others. And so we call it the P site because you can think of a polypeptide. So the P site is the growing polypeptide. The A site is when we only have one amino acid. And then the E site is when empty tRNA exits. And so that's why we call it the E site, the exit site. All right, so that's going to take us to our first practice on slide 22. So you guys can practice looking at your slides and labeling the diagram.